Science and technology and innovation, these are indispensable in the modern economy, in the modern world.
We held a meeting at the MSI, and we resolved among about 6 to 8 of us, local members, first to form PASE Philippines so that we could continue to host meetings here, but also to go on a massive campaign to try to get support from the national government through the legislative and executive branches to invest more in science and technology. So it started with a white paper, a position paper that we co-authored among 6 of us, namely Ernie Pernia, Cesar Saloma, Rador Asansa, Toby Dairita Fataneo, and Alvin Colaba de la Salle, and myself.
So PASE historically, in the past 10, 20 years for sure, have been collaborating with our counterparts here in the Philippines at various universities. And a lot of these interactions probably started as peer-to-peer. But thankfully, we have the Department of Science and Technology here in the Philippines, and they've come up with some really helpful and very impactful programs that actually enable PASE members that are based in the United States and other parts of the world to actually come here and do extensive research and academic partnerships and collaborations.
Well, I think that one of the good things that PASE is doing is really trying to stay in touch with the researchers and the scientists here in the Philippines. So I think that is one of the good programs that they keep on supporting some of the initiatives of our researchers and scientists here in the country, trying to align also their assistance towards the plans of the Philippine government.
It's very important that the PASE members and scientists based in the Philippines are continuously in contact, so we will know the needs of the Filipinos and opportunities that Filipinos can have to do science with their PASE counterparts. So these interactions, such as the conference that we're holding and the annual conferences that we have.
The Philippines and the scientists there have very unique perspective that adds to discoveries and inventions, advancing in general the field.
These members, because they are part of an academy, it implies that they are productive. So they have publications, they have innovations, they have inventions, patents, and they have, they have an impact on society. Okay, so these are the people who can convince government, invest more in science and technology, and that was really the gist of our paper, our position paper in 2000. And it is Ernie Pernia who said that R&D by nature is expensive and it is a public good. So it is important for the government to take the lead in investing in R&D.
To increase the economic growth potential of the country, we cannot continue to just rely on traditional inputs into production as capital and labor. We also need to develop our expertise in science and technology and innovation so that we can come up with new systems, new ways of doing things.
I think that science, researchers, industry, and even the government is really like an ecosystem of knowledge. So I think it doesn't really matter if you're coming from a private institution or a public institution, such as a state university. I think we all have a role to play to improve the research capabilities and scientific knowledge here in the country. I think it is about time for us to realize that one of our roles as academics, as researchers, as scientists, is to actually contribute to that global body of knowledge.
And the role of this cooperation among these government agencies really is to be able to attract science and technology, global-based companies to set up shop here. And that is really key in terms of developing the science and technology innovation ecosystem in the country.
Particularly because there's that deep desire. And what PASE does and can do more of is to promote this relationship between its members.
We really need to ratchet up the appreciation, the recognition of the importance of science and technology and innovation.
And we are all trying to attain what we call the common good, the greater, higher, long-term good of the Filipino people.
Good morning, everybody. I think I am, good morning, Dr. Dalisay. It's an honor to have you in front of us today. Good morning, Jericho, and thank you, Paase, for staging this important presentation for us. I guess it's morning in the Philippines, and it's evening here in America. I see President Mario in the audience and Vice President Completo in the audience. Good morning to all of you. First of all, this is almost the end of Mother's Day here in the United States. And of course, our president has already greeted every one of us, every one of our mothers. And it was Mother's Day yesterday. So I'm just giving my also greetings to the mothers, my mother, all the mothers here in the audience. Folks are still coming in. Folks are still coming in. This is the 20th iteration of expanding the edge of Philippine medicine. And we really are very honored that Paase invited us to be part of this scientific forum. It really is a great honor. Thank you, President Santo Domingo, and Vice President Gladys Completo. My co-host, I am Silverio Cabellion Jr. I live here in Maryland, same as our president, and I chair the research committee of the University of the Philippines Medical Alumni Society in America. I may live here, but my heart is still at home. My co-host today is Dr. Jericho Santos. Dr. Maria Caliante has to have a seminar today, but Dr. Santos, he was one of our first students to come over to the Uniform University of the Sciences, and then he spent a year at the USNIH, and I believe now is back in the Philippines. Jericho, are you with the Department of Biochemistry now?
I'm with the Institute of Clinical Epidemiology in the NIH with Dr. Mela Lapitan.
Wow. Well, Jericho is one of our bright stars in the College of Medicine, and probably a big leader here in science in the years to come. Just watch out. If I may share the screen, Madam Administrator, to introduce our speaker. Let me see. Am I coming through? Hello?
Not yet.
Not yet? Let's see, I should. Okay. Let me see, you are sharing the screen. Okay. Let me see if I can get my slides here. Yes, bear with me a little bit. Bear with me a little bit. Just bear with me. Okay. Let me see.
Dr. Cabellon, would you like to show the bio notes of Dr. Dalisay? Perhaps
Yes,
Feb can help.
I do have I do have the bio already, and that's what I was going to share. Just give me about a minute. Give me just a minute. Hopefully, I can figure this out very quickly.
I can share it, Doc Lily.
Hold on. Hold on, huh? What's going on here?
I can share the
Let me stop the share for a while, and let me just get back from the from the from the very well. Okay, I will just have to just uh, uh, introduce Dr. Dalisay without my slides. I had them prepared. Dr. Dalisay, Doralyn Dalisay, is a cum laude, BS Pharmacy, University of San Agustin. And after that, she went for an MS at the University of the Visayas at Miagao in Iloilo. After that MS, she went to the University of in Australia to get her PhD. And after a PhD in Australia, she went to the United States in San Diego, where she did her postdoctoral degrees there. Somehow she found herself in Vancouver, Canada, and had did research there. And she came near where I was in high school at the Washington State University in Pullman. That would be in the eastern side of Washington State. And then she found herself back in the Philippines in 2015. I wish I could share some of the slides that I made today, but I guess I'm not good enough yet with this Zoom part. So without much ado, Dr. Dalisay, your presentation, please. Thank you.
Thank you very much, Dr. Billy Cabellion, for the kind introduction. And also thank you very much for this opportunity and for the invitation to share what we have here at the University of San Agustin. And also thank you very much for PAASE, the officers of the PAASE, for this opportunity to share our story, what started with University of San Agustin, which is now an emerging research university in the countryside. So the title of this talk is Accelerating Philippine Countryside R&D for the Promotion of Natural Drug Discovery. Before coming to the Philippines, coming back to the Philippines, most of the R&D are in Metro Manila or Manila Center. And we were kind of hesitant of coming home because there's no much of what's going on, particularly in Iloilo City. So before I go on with my story, I would like to introduce you first to University of San Agustin. This is the University of San Agustin is in Iloilo City, and it is commonly or fondly called USA. And it is known for its flagship program in medical technology, pharmacy, and law because of high board exam performance of its graduates. Hence, it has an excellent track record as a primary teaching university. The university was founded in 1904 and named after Agustin of Hippo, the 4th century saint. USA, however, was not a go-to university by researchers for expert consultation and collaboration, even by the local community in Region 6. And it is sad to note that only the active collaboration agreement of USA in 2015, before the Office of Research and Global Relations science was created with its neighbor. However, administration internalization of the country and building activities. USA Tuklas Luna Center, Miles Saludes, who is an expert in chemical biology, drug microscopy, and myself returned to USA from continental USA. And this is where we started to bring research and development in a teaching university in the countryside. In 2015, this is where our laboratories. Sabi ng president namin, wala po tayong laboratory, Dr. Dalisay at Dr. Saludes. So I will give you a small room that you can work on your research. If you look at this and coming from the United States, you will be disappointed, you will be frustrated. And this is how it all began. Kahit isang instrument po doing all these natural products, there is nothing except a very old analytical balance that is not even calibrated for the last 10 years. And so this was a very big challenge for Dr. Saludes and myself in 2015. That's why I told myself I have to go back to the US after 3 months of Balik Scientist, because I cannot do research here in this kind of scenario wherein there is no research laboratory. However, in 2015, since our president was he's a visionary, and he wanted to promote USA not just a teaching university but as a research university, he created the Office of Research and Global Relations in November 2015. So the Balik scientist, Dr. Saludes, and myself joined University of San Agustin in September 2015. And in November 2015, the Office of Research and Global Relations, or we call ORGR, was created. And it was Dr. Saludes who was given a task to be an associate vice president for this. And from 2015, as you will see, the previous slide, that it's just like parang stacker, it was transformed in 2016 July. Not even a year, it was transformed into a research lab wherein the university invested laboratory equipment to seed as a seed grant in order for us to do our research. So the university invested an HPLC, PCR machine, and also other instruments that we will use for our research. And that is the transformation in just less than 1 year. There was already a transformation, and that somehow gave me excitement. Sabi ko, I will give another year for University of San Agustin to really lure me to stay in the university, to help me grow the R&D for drug discovery. And so this laboratory was named as the University of San Agustin Gregor Mendel Research Laboratories. And then it was in 2017 that we seek audience from the president that in order to have a specialized center for drug discovery with impact in health outcomes for Region 6, we need to create a center dedicated for chemical biology and biotechnology, we call a C2B2, and also Center for Natural Drug Discovery and Development, or we call that a CND3. So we were able to get a space in one corner or one area of the university, and this was transformed into C2B2 and CND3. This is how it looks like in 2017. And in 2018, just a year, in less than a year, the C2B2 and CND3 was inaugurated by the former DOST Secretary Fortunato de la Peña, and this is how our USA Research Center looks. We have a foyer here, and then we have a microbiology laboratory, which I'm going to explain later why we have that. And also we have a chemistry laboratory, and also we have a molecular biology laboratory. We have a cell culture laboratory that houses 4 cancer cell lines and 3 mammalian normal cell lines. And also we have our mass spectrometry laboratory. So in this area, and you will see that the support of the USA friars to our vision of creating an R&D in the province or in the countryside, it is there. It is very heartwarming. It nakakataba po ng puso because they really put the investment to put the infrastructure to provide us with a playground. Dahil yun lang talaga kailangan ng scientist eh. We need to have a playground in order to create our research. And the USA administrators and the friars provided us with that infrastructure. And of course, the infrastructure is nothing without the facilities. That's where the DOST partnered with USA because they have seen that USA is investing on the infrastructure to house different types of facilities and equipment. And so DOST partnered with us, and they've awarded us with several grants on drug discovery and development. So we also have our research meeting room where we do all of our discussion every time. We have a progress report and progress meeting for our different projects. Another expansion of research infrastructure was done during the height of pandemic. There was this big infrastructure to expand C2B2 and CND3 to house the first of the facilities in the Philippines. And that is the multi-mass spectrometry laboratory that was started in May 2020, and the NMR laboratory that houses the first cryoprobe NMR in the Philippines. This was in August 2021. And so you will see here that the USA administrators really was looking into making USA as the hub of the R&D for natural drug discovery and development in Region 6. And so therefore they were able to put a lot of investment in the infrastructure. So take note that this was done in 2020 and 2021 during the height of pandemic. Kaya nga sabi ng mga visitors namin, meron bang pandemic sa Iloilo? Bakit there is a lot of buildings and infrastructures going on despite there is a pandemic? And this shows and demonstrate that the university is really the support is so strong that the investment that they put into will have an impact, not only on the university itself for research achievements, but also impact in Region 6, particularly in the economic aspect, which I'm going to discuss later in my talk. And so in 2022, there is an inauguration of the first cryoprobe NMR laboratory and the multi-mass spectrometry imaging laboratory by the former DOST Secretary Fortunato de la Peña. So what are the different types of programs that we work at USA that has health impact? First, we have what we call as informatics for health, wherein this is more of data mining and using informatics to bring health sector or health information to the community. And this is under the Center for Informatics, which, by the way, is directed by a Balik scientist, Dr. Romulo de Castro. We also have the functional food. We also have the Center for Heritage and Indigenous Culture, because the university wanted to not only looking at the health, the discovery, drug discovery, but also look into heritage and indigenous culture, because that is also important to health, because there are some indigenous treatments used by the indigenous people in their treatment of different types of diseases. And so we partner with SHIC, or the Center for Heritage and Indigenous Culture, in other areas of our drug discovery projects. We also have the functional foods and marine biotechnology, which is now covered by CND3 and then C2B2, which, by the way, is directed by yours truly for the Center for Chemical Biology and Biotechnology, and Dr. Saludes for Center for Natural Drug Discovery and Development. And also we have peptides in drug diagnostics and drug delivery, which is currently an undergoing program in collaboration with Tsinghua University, the first university in Asia in 2020, the best university in Asia in 2021. And we're looking into creating a lab in a chip in detecting different types of diseases, particularly autoimmune diseases such as lupus. So our laboratory at the University of San Agustin, particularly C2B2 and CND3, we considered this as a FIFCO. It's a fully integrated pharmaceutical company, as we call, because everything from the raw material until determining the structure of the compound, until determining if the compound is toxic or not, everything is integrated in this center. And so at the end of the day, we were able to identify leads and not to wait from one lab to another lab to tell us what's going on. And so there is a very dynamic and collaborative aspect when it comes to the FIFCO projects that we have in the lab. So this is our 2+ Luna Center, the drug discovery program, from discovery to preclinical studies. So our focus is on marine microorganisms at this moment, because I'm going to tell others later. So we have a long list of strain collection. We have the isolation and the identification of these microbes. We do our fermentation and scale up production here, and we do whole genome sequencing and a lot of genome mining, looking at the biosynthetic gene clusters to see any unique compounds or any unique biosynthetic gene cluster, and then merge them together to identify, is this a new scaffold, or is this a new analog? How is this analog produced, and how is this new scaffold is being produced in a biosynthetic way? And so aside from microbiology, the microbiology are all of our strain collection undergoes different types of screening. The anti-cancer screening, we have colon cancer, ovarian cancer, breast cancer, lung cancer, and prostate cancer that we do our screening here. Antibacterial screening in all escape pathogens, and these are all certified multi-drug resistant organisms. And we also do the secondary and functional screening, meaning we have to know whether it is very specific in that target, or it is broad when it comes to its different types of activity. We also have our chemistry. The chemistry do a lot of the extract library, the analytical chemistry. Before we venture on the isolation of the compound, we do a lot of the replication, because there are two things that we have in mind. Are we looking for a new scaffold, or are we looking for another analog or a new compound, but yet the activity is not yet reported? And then we do structure elucidation here in USA. We are also doing a lot of metabolomics, because we have a different platforms for mass spectrometry in the lab. We do a lot of bioanalytics and also biomarker discovery using our multi-imaging mass spectrometry. And in the end, we have this mantra that we have to know whether we call it fail now than fail later. Because we do not want to introduce a compound to preclinical trial or clinical trials that are already toxic. That's why we have a toxicity testing here in C2V2 and CNV3. We do target validation, the mechanism of action, and we are part of the ADMETOX program of the PCHRD. So really the inspiration of why we're working on microbes is that we wanted to revive the erythromycin. It's so inspiring because the soil where the organism comes from that produces this erythromycin comes from Iloilo. And being an Ilongga myself, I want to do another venture on that. And the inspiration of erythromycin is the one that drives us to find new scaffolds or new antibiotics that someday we will be able to provide an antibiotic against escape pathogens that is owned or discovered in the Philippines by Filipino scientists and also for the good of the Filipino people. And that is one that inspiration drives us every day. And so with that, if erythromycin came from an organism isolated from a terrestrial soil, our collection is different this time. We're looking at the marine sediments under the seafloor, because there's a lot of things going on already on the soil terrestrial for organisms for drug discovery. So we look into the Philippine marine sediments, or tinatawag namin itong bacteria sa buhangin. And it was in 2017 and 2018 when we received a grant from the National Research Council of the Philippines and also DOST to look into the bacteria sa buhangin, and it enabled us to do expeditions all over the Philippines, from Batanes to Mindanao, and in some remote areas, including to Bataha Reefs. So I'm going to bring you to this short video. So this is the University of San Agustin, a very small university, landlocked university, the heart of the city of Iloilo. And so this is the C2B2 and CNV3. And so I will bring you to one of our expedition sites. This is in Tubataha Reefs in Palawan in 2017. We were there for 6 days and 7 nights in the middle of Sulu Sea. And this was the research vessel that we were able to house our laboratory. And I am the only woman in the expedition during this expedition, and we are a team of different divers and also underwater photographer. So we use a sediment core sampler. Back in the States, when I was doing this, the sediment core sampler that we use in UCSD and also in University of British Columbia are very long, like heavy, mechanized scoring system that we have. But back in the Philippines, sabi ko, paano kaya natin gagawin ito? So I drawn and partner with another mechanical engineer to work with this sediment core sampler. That's why makikita nyo that we have that improvised sediment core sampler, because I'm interested at looking at 1 meter below the sea. So in 2017, we went to Apo Island in Negros Occidental, in Balikasag, Bohol, Batbatan Island in Antique, the Nugan Island in Negros Occidental. We also went near Boracay Island, but on the backside, in Crocodile Island in Aklan. We also went to Mararison Island in Antique, Nogas Island in Antique, facing the Sulu Sea, Olotayan Island, Capiz, Seco Island in Antique, also facing the Sulu Sea, Sumilan Island in Cebu, and also in Unisan Island in Guimaras. So this was in the first year, 2017. And we continue on with our 2018 expedition, wherein we went to outside the central and western Visayas. We went to areas including Apo Reef, Occidental Mindoro. Unfortunately, it has oil spills right now. There, we also went to Batanes Islands to do the sampling, to Quintinua Island in Camarines Norte. We also went to the eastern part of the Visayas, Kalangaman, Leyte, also in Siargao area, in La Jañosa Island, Mantigue Island in Camiguin. Also, we went to Pujada Island in Davao Oriental. That's the farthest we went down south. We also went to Cuatro Island, Leyte, and White Island in Camiguin, and of course, the Tubataha Reefs National. So, of these different core samples that we have, which until now we have a library of core samples in 2017. We did not dispose those because it's a very rich biological sample that we could get a lot of information out of it. But nonetheless, out of that sediments, out of that buhangin, nakakuha po kami ng more than 3,000 of these pure isolates. I have not seen such diversity of this when I compare this work with my work at UC San Diego and University of British Columbia in Canada. And it is so diverse that they produce these different types of diffusible pigments and also different types of morphology. So we consider this as a drug discovery microbial biobank in the Philippines, which currently we're looking at antibiotics, anti-cancer, and currently we're working on anti-SARS-CoV-2 inhibitors. We are working to collaborating with anyone who has expertise for dengue, tuberculosis, and malaria. And we are just waiting for someone, any group here in the Philippines that could collaborate with us and use this drug discovery microbial biobank to look for these inhibitors against these different types of tropical diseases. So what do we do here in Natural Products Drug Discovery? Aside from the in vitro bioassay that we do in-house, we do a lot of genomics and metabolomics, and this is the one that drives our isolation and purification. Aside from bioassay guided purification, we are also guided with metabolomics and genomics. That's why it is a very multidisciplinary type of natural products discovery, and we are very blessed to have all these instruments and facilities to help us identify that different types of compounds with different types of activity. So this is one of our publication in scientific reports by Nature Publishing company. So it was published in 2021, and we already received 16 citations of this. And I am very happy because globally, our work has been identified. And in fact, many laboratories in Europe and also in Asia is sending emails to me to do some collaboration because they have found that what we are doing here is currently very relevant. And that is also one of the major programs in Europe right now in looking at this bacteria sa buhangin. So what you have seen here is that we are not only creating publication. Look at these young individuals who are the authors of this, which by the way, lahat po ito taga Region 6. These authors, this is all Filipino authors, lahat taga probinsya, and and graduates of universities here in Iloilo. And so with this, some have already left the lab for their PhD because we are also encouraging them to do graduate degree outside the Philippines and to come back and being inspired of what we also do here so that they will come back in the future to create or join C2B2 and CM. But nonetheless, this scientific reports paper looked into 11 sites of collection here in Central and Western Visayas. And we have identified that the most number of isolates that we recovered was from the Nugan Island. The Nugan Island is a protected habitat in Negros Occidental. And we have seen a lot of novel species there and very diverse, which also correlates with other sites where we collected the isolates, because those sites that are protected, meaning they have their own local government unit that protects the area. There is an active PUMB. We have found that there is more diversity and a lot of novel species that are present in that area. And so on that area alone, the 11 sites alone, we screened about 2,212 of this against S. aureus, E. coli, and Pseudomonas aeruginosa. And after the first initial screening of about 2,000, we were able to narrow it down to 218 lead isolates. And from there, it underwent another secondary screening to get all these false positive results. And we have this 92 confirmatory isolates that we considered as antibiotic and anti-cancer producing compounds. So this is kind of a busy slide, but I would like to point you to these different types of species of actinomycetes that we were able to isolate. So some of this have a big population of one single species, as shown in here as Streptomyces parvulus, and also in this area. But if you look into this branch, this is where the novel species are found that we currently are working into mining the metabolome and also mining the genome of these different types of species. If you look at this circle outside this tree, this circle has a heat map. This is the bioactivities based on the antibiotics and anti-cancer. The very pink one shows that it is a very active antibiotic and anti-cancer producing isolates. The green one shows that there is no. And so you will see that even they can be of the same species, they can also show different types of bioactivities. And that is one highlight of this paper that we emphasize, that we should not take for granted a strain of the same species of organism, because they have a different metabolism, as shown not only by their metabolites but also by the biosynthetic gene clusters that you can see here by circles and triangles and squares that dictates this bioactivity that they produce. Another one is our marine drugs paper. So we look into how a group of organisms from area here in Iloilo have a different activities and also biosynthetic gene clusters. So this was cited in 2021, and we currently have 8 citations received for this paper. And so we looked at 3 different gene clusters for the biosynthetic gene clusters. So we have the PKS1 and PKS2, and also we have the NRPS, because we are interested at looking at all of these different types of modules that creates these different types of activities. And our hypothesis here really is that are all streptomyces that has PKS1 or PKS2 or NRPS are directly correlated for its bioactivity. And we have noted that here we have only this cluster 2 that has PKS1, PKS2, and NRPS that has bioactivity. If you look into cluster 1 has PKS1 and also PKS2 and NRPS, yet they do not have activity. Cluster 3 here, they have NRPS and PKS2, and yet they have bioactivity. Also here in cluster 4, they have both the PKS1 and PKS2, but it has no bioactivity. And so therefore, we are currently working on the metabolomics on this, and we have a paper in our pipeline to describe what's going on in this type of different isolates or extracts that produces these compounds that are directly correlated for its bioactivity. This paper was published in Frontiers in Microbiology. We wrote this at the height of the pandemic. I still remember the lab was closed for 3 months. And so to make use of our time, we wrote 3 papers actually during the height of the pandemic, and were able to publish one of these papers in Frontiers in Microbiology, and it has received 13 citations at the moment. So we have found here a newly identified anthracycline shunt metabolites that destroys the cell membrane of a multi-drug resistant Staph aureus. And again, these are very young researchers who are co-authored in this paper. So when we are looking at this, the first assay that we did was to simply measure the biomolecules that comes out of the cells after a treatment of antibiotic. We're looking into the protein that have leaked out the cell, and particularly also the DNA. And we were able to look into, there is something going on. And during that experiment, I said, oh, this is something promising, because the protein and the DNA values that have leaked out of the cell is there. And perhaps it has something, the antibiotics is targeting the cell membrane. And during the time, I was not convinced of the figure A, because this is just a spectrophotometric measurement. And then we looked into the transmission electron microscope. And if you look into the untreated, the cells are round, and the membrane is still intact. And also the vehicle that was used, the DMSO that was used for dissolving the antibiotic, they are happy. And in fact, some of them already dividing. But here, the antibiotics, the cells treated with antibiotics, they form an aggregate and they are collapsed. And when I saw this, I was so convinced that yes, there is a membrane integration mechanism that these antibiotics work on. And so that's the reason why we have found that there is protein that have leaked out and these biomolecules that leaked out and form into this clump or aggregates of cells. And so these are the two anthracycline antibiotics that we found. This was first report of these antibiotics or natural products because these are shunt metabolites that are only produced when you manipulate a streptomyces, genetically manipulate a streptomyces organism to produce different types of antibodies. But in a wild type, you are able to identify, you're able to isolate this. And so the pharmacophore for this is these 4 rings. We call this as the anthracycline. And one has a 6.25 microgram per ml MIC. The other one has 50 microgram per ml MIC. And if you look at the difference between the two, it is in ring A that this has an hydroxyl functional group, and this somehow affected and influenced the minimum inhibitory concentration, and so that somehow weakened the activity of the antibiotic. So this is very interesting that this functionality weakens the antibiotic activity of this antibiotics. Another one we have here, a journal for marine science and engineering that we also wrote during the height of the pandemic. It has received already 17 citations, and this came from Iloilo marine sediments. Buhangin, bacteria sa buhangin po ito ng Philippine. So this comes from our collection from Islas Gigantes in Iloilo. That's how it looks like. This organism is a novel species because this is entirely different from its own, from several streptomyces, neighboring streptomyces species. And during the time, we look into the PKS, PKS2, and RPS, and you will see that PKS2 amplified gene. There is this amplified gene PKS2 and also NRPS, but not in PKS. And that really is one that drives us to look into what is this PKS2 molecule that is responsible for its antibiotic activity. And these are the type 2 polyketide antibiotics that we found. It is an antibiotic, anglocycline glycoside. So previously you have 4 rings there as a pharmacophore. Now we have 3 rings as its pharmacophore. And the only difference here is the sugar moiety attached to this anglocycline anglocycline pharmacophore. And you see that if you have one glucose attached, a sugar attached here, it has a very low MIC or low bioactivity. But if it has 3, it has a very high, somehow not high, but 62.5 microgram per ml. And we have some papers to publish soon on the different types of analogs and structure activity relationship of how anglocycline glycoside targets different types of escape pathogens. And the latest discovery that we have here that we published in microbiology spectrum, which we were very happy that this was accepted in a high impact journal. And we currently have 2 citations from this paper. We have a new species, bacterial species, that we isolated from Tubataha. Therefore, we named this as Streptomyces tubatahanensis. And last week I was interviewed by BBC Asia that they have the plan to feature this type of expeditions that we have in Tubataha, and that is still under discussion with BBC Asia to look into the marine ecosystem here in the Philippines, looking for different types of organisms thriving on the marine sediments. So this is how the DSD 3025 looks like. And the whole genome mining is really look into these different types of biosynthetic gene cluster. And in fact, it contains one major enzyme that is capable of attaching a halogen, either chlorine or bromine, to any scaffold that 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 the enzyme halogenase can work into. And so we were able to look or find these antibiotic and anti-cancer carbazole alkaloids. It was so unfortunate that when we discovered this, the Chinese laboratory or Chinese group uh, did a publication 2 months ahead of us. And so I said, well, there is no prize for a second place, but nonetheless, what we put into the body of knowledge is that the Chinese scientists or that paper, they were not able to identify how these compounds, these antibiotic and anti-cancer compounds, are biosynthesized. Even though they have claimed that they did a biosynthetic, they did a whole genome sequencing, and they just wrote in the paper that the biosynthesis of these carbazole alkaloids followed a novel biosynthetic pathway. And so in this paper, we revealed to them the biosynthetic gene cluster, and one of the corresponding authors of that paper emailed me that they were very thankful to that a paper came out after their paper looking into the biosynthetic gene cluster of this carbazole alkaloids. And I'm just really glad that, you know, from 2015, from that very storeroom type of lab that we have, we can now produce publications that are globally recognized, and scientists from all over the globe are sending collaboration or email and look into different type of aspect on this discovery. So what we're looking right now is this microbial biobank for drug discovery. So we have our grants from the DOST PCHRD for 2+1 program for antibiotics and anti-cancer compounds. We currently are looking for beta-lactamase inhibitors, particularly for those targets that produces beta-lactamase that crunch or eat the beta-lactam antibiotics, making these antibiotics not effective anymore. So we have beta-lactamase inhibitors project that we're looking into, and also SARS-CoV-2 3CL-Pro and PL-Pro inhibitors. These two projects are funded by the DOST and RC. So for the SARS-CoV-2, we're looking into the very specific, target-specific, this is not cell-based, target-specific, the enzyme, a recombinant enzyme of the main proteases or the 3CL-Pro and the PL-Pro, because these are enzymes that do not mutate. Whenever there is a chance of mutation, unlike the spike protein, that there is frequent generation of mutants. That's why we have a different variants, and targeting that for drug discovery is not really advisable. So we look into a target that does not mutate and has no homology with the human proteins. And this is what we're looking into, the main proteases and the PL-Pro. And we already have hits on this. We have 32 out of 81 were found to be active against SARS-CoV-2. 4 out of 81 were active against PL Pro, and we are looking into the IC50 and now looking into the metabolomics and genomics for the identification of these small molecules. And these are types of its IC50. You will see that there is about 2.5 microgram per ml of the extract, and you will see that this is very promising to look into this SARS-CoV-2. One thing that I would like to emphasize is what we're doing here is not just an academic exercise to look or to produce publications for university compliance, but one of our major work here in University of San Agustin is our active partnership with industry. We have currently 4 active industry partners for functional food. Herbanex, which is located here in Region 6 in Negros Occidental. Mariden and Galen X, which is located here in Iloilo. These are local pharmaceutical company, and just it's a startup company. The Galen X is actually a sister company of Mariden that was started in 2015 when we joined University of San Agustin. That's why from the founding of Galen X and our joining in University of San Agustin, it helped Galen X to produce innovative products, which I'm going to present later on. And also we have a current outstanding project with Del Monte Philippines, looking into different types of problems and also putting added value to their product. Our project in Herbanex right now, we call it academic industry government collaboration, because this is funded by Cradle, DOST, PCHRD. We now have 2 products, that the Tawa Tawa effervescent tablets, which is used for dengue, is currently in clinical trial. One here in Iloilo in collaboration with the TMC, the Medical City of Iloilo. The turmeric latte blend, we were able to identify the first peptide that is present in turmeric that is highly antioxidant, and it was formulated for this turmeric latte blend. This will be released in the second quarter of this year, and it will be available in the market. And this is with laboratory or Herbanex laboratories. One is also with Mariden. This is looking into honey. This is, by the way, funded by the USAID. And with this, we were able to look into one particular component in Philippine honey that has a wound healing effect. And this was formulated by Galen X into a formulation of a wound gel, and it was validated by also a publication that we have published here. And this product is currently available in Shopee and Lazada. And if you are going to follow Galen X, a social media account, there are so many testimonies, particularly in diabetic foot, that were healed by this Hanel wound gel. And we are very proud that this has already, a lot of products already have been sold, and there's an economic impact of this in Region 6. And already Galen X has started to import this, export this product in Singapore, because there are also already Singaporean users of this. The next product that we will launch on July 13, 2013 is a PCHRD project on a bean from Region 6, we call it Cajos. This is a formulation that was formulated by Galen X for antibiotics, whether it is for cosmetic wound infection, it has activity, and we have a paper to describe these different types of molecules that they produce, and the formulation will be have already registered in FDA and will be available for in the market after the launch on July 13, 2023. So this is one of our major big company that we are in partnership right now in Del Monte, Philippines, because they want to add more value, not just nutrition. They're looking into what other functional components there is in order that they could add into the value of their product. And so with this, we have invention on the compositions to lower cholesterol that we already filed with IPOFIL, and we already did the review has already been done. We have a response to the review for IPOFIL. We also filed a PCT for international patent in Singapore, Japan, and United Arab Emirates. And we have several utility models. And so this is the major paper that we released recently for this project, looking into which part of the pineapple there is a high concentration of a cholesterol lowering compound. And we have found that it is in the area near the peel. And so that's where these compounds are concentrated or spatially distributed. And we were able to identify that using multi-imaging mass spectrometry. So since 2020, in 2015 to 2019, wala po talagang paper na lumabas. I sabi ko, my publication track was drying up since bumalik kami ng 2015. But I said, let's just do research, and lumabas yung first paper in 2020, and since 2020 until this time, we have 18 publications and counting, which I'm very happy. So these are some of the Q1 and Q2 journals that we published our publications here in C2B2 and CND3. Our patents since 2020, we have 6 patents in the Philippines, 3 in PCT, 6 registered utility models and trademarks, and still counting. We have here, when it comes to prestige, 15 international awards, 22 national awards, and 6 regional awards, both on the project, the principal investigators, and the research staff, who are part of the empowerment and the capacity building in Region 6 for the next generation of drug discovery and development researchers. So since 2020, we hosted 7 PhD scholars, 19 MS students, and 32 Bachelor of Science students in pharmacy, medical technology, chemistry, biology, chemical engineering, biological engineering. We also have our academic partners. In 2015, isa lang po yung partners namin, yung CIFDEC. But now we have several partners all over Asia and other parts of the world, in US also, and also in Malaysia, in Indonesia, and in Taiwan and in Japan. And these are our industry partners, because that's one thing that we have seen here in the Philippines. Academic institutions are not into collaboration with industry. And so this is one thing that we are very happy that industry partners are industries, particularly the startups and those who have innovative ideas that they want to put value into what they produce in the product. So they contact us and partner with us in that aspect. So we have for the external grant in 2016, we have zero. There's no external funding before. But now, as October 2020, we have now 15 external funding that we receive from DOST, from global institutions that also giving external funding for drug discovery, other aspects of research here in US. So one of the public service of University of San Agustin, or USA, was during the height of pandemic. Region 6 has no testing lab for COVID-19. And so USA, particularly with C2B2 and CND3, hosted the training of a training ground for molecular biology for medtechs and also from pathologists to establish COVID-19 testing lab in 5 hospitals and 1 city health office here in Western Visayas. And these cross traditional, professional, and institutional boundaries in order to communally mitigate the impact of the pandemic. And so here are the different hospitals who were trained here in C2B2 and CNB3. And the training is not complete without partnership with the Philippine Genome Center Visayas and also the Philippine Science High School, where they have the certified bio security officer that are certified in the Philippines. And so this is one of the public service of USA, and we are very happy that with this, there are now many COVID testing laboratories here in Region 6. So I would like to thank the Balik Scientists Program and also the National Research Council of the Philippines, the Philippine Council for Health Research and Development, particularly with the Duplas Lunas Development Program, and the DOST DOST Department of Science and Technology for the unwavering support. You know, kahit ano kagaling ng isang scientist, if it's not supported by the host institution and not supported by the funding agency or the government agency, wala pong mayayari. Kahit Nobel Prize pa yan siya. So this is something that I would like to emphasize. The reason why you are able to have this kind of research R&D ecosystem in Region 6 is because of the tripartite relationship. The scientists, the host institutions, the funding agencies, and a new partner industry. So these are the 4 factors that you have to look into when creating an ecosystem sa probinsya. Because this is where you are going to thrive and to make a bigger impact, not only in the university aspect, but also in the community, in Region 6. And hopefully what we are doing here will have a more impact in the whole Philippines. So this, I would like also to thank our scuba diver team who helped us with the selection of the sediments, and also the LGU who gave us the prior informed consent, the gratuity permit in order to collect these different types of sediments. And also these are the new generations of the drug discovery and development researchers in Region 6. So they are from Region 6, and we are very happy that we are empowering them to do such work that has a health outcome and impact in the economy in the regions. So please follow us in our Facebook and Instagram and Twitter accounts. And you can email us at C2B2 and CND3 if you have some questions or you want to collaborate. So thank you for your time.
Thank you very much, Dr. Dalisay. That was really a treat to hear you this morning, evening here. Before I turn over the question and answer period to Dr. Jericho Santos, I would be remiss if I did not recognize Dr. Giselle Concepcion, or else this presentation would not have happened this evening, because on April 23rd, Dr. Concepcion sent an email to PAASE citing the paper with regards to, excuse me if I cannot pronounce right, the actinomycetes and the chemical carbamazole as antibiotic and anti-cancer. So that is why we are here tonight, because of Dr. Concepcion's email. I have to tell you that the Marine Science Institute also has about 600 molecules, biomolecules, that they have in their library waiting to be developed, I believe, as antibiotic and anti-cancer agents. They are from sponges and mollusks. And I see that the material for the studies at Iloilo, San Agustin are from the sediments. So I have to tell you how inspired I am in hearing the presentation of Dr. Dalisay in front of the Dean of the College of Medicine. I said I was jealous of Singapore because we don't have anything like what Singapore does. But I have to tell you now that I'm jealous of University of San Agustin, coming from the University of the Philippines, that they do have, I think, a very rigorous setup in trying to find drugs against cancer, against, I see there that there's a note on SARS, on COVID-19. So before I start getting too excited and talk all evening, Jericho, Dr. Santos, your turn, sir, to field the questions. Oh, you have to unmute. Okay.
Hello, good afternoon. Can you hear me?
Yes.
Okay. So you would like to open the floor for questions. So anyone who would like to ask some questions. There is one question here in the chat box from Dr. Primavera. Thank you for a great presentation, Dr. Dalisay. As a marine ecologist, I was struck by your finding that Dunjugan has the highest diversity in your isolates. I have found the same diversity in beach forest species in the 3 times I did field work. Just for the record, protection in Dunjugan started with the NGO community, with some 4 to 5 national and international NGOs, I think. And NGOs purchased half of the land, and this is how protection came about. It is not part of the government. So PAS, at least not in 2018 when I was last there. Based on your findings in Dunjugan, what recommendations could you send to DNR about protection of the marine habitats, especially the sediment habitats, aka soft-bottom habitats, in addition to the more popular mangroves, seagrasses, and coral reefs?
Thank you very much, Dr. Primavera, for that question. Yes, the Nugan Island is now owned by not owned by the government, but it is protected by an NGO. And after our study, we went back to the Nugan because they wanted to know what we had found. And based on our findings, yun nga, meron tayong maraming diversity. And it is true that the marine ecosystem, the more diverse, there is a direct correlation on the bacteria sa buhangin. And the more marine grass or mangroves or coral reefs there is, marami pong diversity. That is also true for Apo Island and Tubataha Reefs National Park, because these are protected area and yung marine ecosystem is so diverse. And yun nga, meron talagang correlation on the organism, not only on the coral area, but doon din po sa buhangin. So I would recommend that to increase the ecosystem, the diversity of the ecosystem in the area, to create more diversity, not only in the macro level, but into a microbial aspect, where we can use these microorganisms not just for drug discovery, but also for industries as a source of enzymes, source of pigments, source of food later in the future.
Dr. Santos,
Yes.
can I follow up?
Yes,
Yes.
yes.
Thank you, Dr. Dalisay. Ang thinking ko, no, protected areas focus on terrestrial sites. Whereas sa Danhugan and the other islands you mentioned, mga maliit lang sila, no? And it would be ideal. I dream of each big island complex having protected, a small protected, 100% protected, no, from ridge down to the pinaka na sediment, so we get all the diversity that you discovered. Now, as far as I know, Danhugan lang ang ganun, and it's not even government, it's private initiative, no? Now, I'm thinking, since the Mandanas Garcia Supreme Court ruling came down last year, giving LGUs up to 1 trillion pesos, no, of funds. Pwede bang the bigger cities, the bigger towns could purchase one small island and really protect it 100%, so that we have, we have these ideal sites for beach forest, for lowland forest, down to the sediments. So pwede ba could you make such recommendation yourself, or SANAG, or even PAASE? Kasi yung Mandanas Garcia ruling, no, hindi pa na-implement, and the national government is really trying to hold on. But that's Supreme Court. They cannot hold on forever. So if we are proactive, tayo sa scientific community, and tell the LGUs, this is how you can spend your trillions of pesos, baka mas okay siya. Thank you, Dr. Dalisay.
Yes, so thank you, Dr. Primavera, for that comment. Yeah, I think we really need to be very proactive in influencing the LGU on that aspect. Dahil hindi kasi nila alam there is no scientific basis. Why should they protect their environment? So hindi nila maintindihan. So perhaps this is something, PAASE could also lead this undertaking, advocacy, to talk to LGU. But what we did in that 23 namin na site, we really went back to the LGU to tell them this is what we found, and this is the diversity that we've seen on the markets. And yun nga sabi nila, yung municipality, nila, they are going to put that sa resolution to do a protective kind of protection on that particular aspect. Ang alam ko lang na merong resolution that was released after our report to them was the Kulase Antique. They were able to put that in their resolution to protect one island that we went for the expedition, particularly with the Seco Island, but the rest hindi po namin. So hindi na namin na follow. So perhaps this is one area siguro sa marine scientists in sa Pilipinas to talk to our LGU in order to have a protection for these different types of islands, pristine islands.
One step further, hindi lang talk to LGU and they have an ordinance. Wala yun. They buy it, they purchase it. Kasi when they have the title to that, they can kick everybody out. We have this example sa Leganes, Iloilo. The title to a mangrove, an abandoned pond area, is held by the LGU. So talagang, it's not just an ordinance. Iba na yung you own it. In the Philippines, theoretically, dapat law should be upheld. Pero we know naman na hindi. So, yun lang, nabilhin nila using the Mandanas funds. Thank you.
Thank you. I'll just read the next question here in the chat box and feel free to chime in after I've read the question to follow up. From Dr. Villalobos, thank you, Dr. Doralyn, for your comprehensive presentation. With all these compounds you're studying that may be potential antibiotic and anti-cancer drugs, is there somebody doing some business development? It's difficult to do development work on so many entities at the same time.
Thank you very much for the question, Dr. Villalobos. One event that we will be doing this year will be to invite investors and also startup companies to take what our technology into the next level. So yung mga pharma companies in the Philippines, we are going to invite them, and this is in partnership with PCHR. So pinag-usapan na po namin ito. How are we going to have the next step? Dahil developing it into a drug is not our expertise. So somehow another entity or another agency will pick up the technology from us and develop it into a drug. So yes, that aspect of inviting these different agencies on drug development will happen within this year. And we have our business development person on drug discovery. He is our currently director of our ITSU. He's British, but he is working in the university at the moment, and he has background in drug development aspect in UK, and he's the one that helps us with the business development of our.
Thank you very much, Dr. Dalisay, for that answer. It's good that you are really thinking of business development, because you cannot just stick to drug discovery like that and so on. And usually the business development is run by business people and then technical advice from scientists. Thank you.
So we have another question here. I think this is very much related to Dr. Cabelon. You were saying something.
Dr. Santos, if I may be given the opportunity to question, it's corollary to the question of Dr. Villalobos. So now we have demonstrated that there are some actinomyces that could be anti-cancer and anti. So the next step is, I guess, maybe a phase 1, and later on a phase 2, and then later on a phase 3. So you have to go through all of that before, I guess, the business development. What structure do you have in the University of San Agustin to follow that up, to follow that perhaps into a phase 1? I noticed that you have some partnership with some hospitals, and then later on get them into patients. But is there some thinking on how it goes further into the clinical development as Dr. Villalobos has alluded to?
Yeah. So thank you for that question, Dr. Capello. So, so what we have here in, at the end of the day, we will have a compound that we know that it is effective. We know that it is not toxic, and we know that it can go to, an investigational drug. So in our platform, after our discovery here, it will undergo some orthogonal testing, which we will be done in collaboration with NIMBB under the lab of Dr. Garcia. And then after that, we will do the ADMET, the absorption, distribution, excretion, and toxicity program. And it is there. After that, then we will know whether we could do the clinical trial 1, 2, and 3. Do we have a program of that here in USA? We don't. That's why we have to partner with an agency that could help us take that product or that compound into a level for clinical trial 1, 2, or 3. So we really need help because hindi po kasi yan expertise po namin. Business side, we are not businessmen, so we need also help on that. So PCHRD is already already know where we are at, and they because of these promising compounds that we have, they are ready to help us connect with some agencies that can push this into the level of different types of clinical trials should we have a compound to move to these clinical trials.
This is Anne Villalobos. Can I make a comment? Usually business development is done early on. That is drug discovery, and then before going into any development work, because you need to focus as to which one to develop. It's so expensive if you have so many at the same time. And at the same time, you have your manpower to be put in there and so on. And the important one in business development is after you have done all this one and so on, will you have a market? Can you compete in the market? If you are just duplicating an inferior product, it's not worth it. So that has to be done early on, the business development. So if you have thousands of compounds or something like that in your library, you cannot develop them all. They may just be overlapping in terms of activity or function. So you need to have that business development early on.
Yes. So yeah, that is
Yeah.
correct. Yeah,
Okay.
that is correct. Yeah.
I've been involved in Johnson & Johnson for
Yeah,
business development.
And also there is a need there to have a supply chain. Can you supply? Yes.
Yeah,
So
that
those are the things that we have to think about. Yes. So our ITSO director, Dr. Baghdadi, is the one who looked into this aspect because he's into drug development in UK, and he has already plans on doing this different business aspect that we have here, or possible products that we have in the market. That's why the comment of Dr. Villalobos is really very important, that business development is early on during the drug discovery. That's why the reason why our partnership with Galen X and Herbanet is successful because from any project, we already talk about supply chain. How are we going to market this? What will be the product? How are we going to move this forward? That somehow is very successful. On the side of the basic science that we have here in the University of San Agustin, we were starting in a basic science research, hoping that it will be taken from us for drug development. So, which we thought easy, but it's not easy. That's why we really need help from the business, private sectors, and government sectors. Should we have a product that we are going to take in the market, because this will be something that not within our expertise anymore. Thank you.
Dr. Concepcion, you're raising your hand.
Thank you, Jericho. Once again, never-ending applause, Doralyn. You're doing in a few years what we've been trying to do for more than a decade. And one reason you are successful is because you are from a private university. And in UP, Dr. Cabellon, we know that we are retarded or hindered by partnering with private companies and also with procurement. But nonetheless, let's just keep hopeful. In our case, to clarify, in MSI, we have 6,000 microbial isolates. Some are similar to Doralyn's, but they have a different, they are symbionts of marine invertebrates. And then we also have compounds, but I don't want to talk about my research and those who are the new leaders. I think we should focus on Doralyn's because we need to have a first success story. And I just wanted to carry on the concern of Dr. Ann Villalobos, which is towards commercialization. And we always think we cannot put the cart before the horse. And after we have shown molecular targets and cellular activity, targeting cells or viruses in cells, then we have to do the in vivo model, small animal model, to demonstrate proof of principle or proof of concept. And we have one model that we are developing now for cancer. And then, of course, it's now aided by all these instruments that the DOST provided Doralyn, including, in situ mass spec to look at the metabolites in the organs, like the liver and the kidney, Dr. Cabellon, to demonstrate if there is nephro or hepatotoxicity. But then again, the bottom line is what is the efficacy and what is the selectivity, the efficacy. So we're always looking at very potent compounds. And, for antimicrobial, which is a big challenge to commercialize all over the world, because aside from COVID and other emerging viral infections, the big pharma companies do not really want to invest in antibiotics. So that's really why there is drying up of the antibiotic drug pipeline. And some time back, we were made aware that the EU was supporting a program where if you could show 30 micrograms per mil concentration dose active in an animal model, like a mouse model, then they could find support for you. And then they even increased it to 60 micrograms per mil in vivo. But that does not correlate with in vitro MICs or IC50s, say, for cancer. So there are many other factors that come into play when you do in vivo. So, it's tough to get that small animal model. And with that, if you really showed significant activity, because you had a compound that was active for antimicrobial activity at single-digit micromolar or maybe a low tens micromolar concentration, then it would probably correlate with good in vivo activity, but you won't know until you try it. Okay, so of course there are ways to model this, and that is why our member Dan Tagle is very much involved in organ on a chip model for 3D semi in vivo testing, but we don't have access to it. I had asked him about it, and he said they're fanning it out through private companies. US NIH is doing that. So maybe we should target those types of companies as partners. But anyway, going back to the level of activity, which is very important. Well, I've been told by some companies, including those in the US, that if you are able to show, say, anti-cancer in vivo activity, then they would be willing to look, say, at developing it as an ADC, antibody drug conjugate, if you're able to conjugate it with an antibody. Okay, so that it's targeted therapy. So things like this. But anyway, bottom line is, Doralyn, I know you are very concerned about looking for novelty in the chemical scaffolds, And I am curious about your patents, whether they're composition of matter, because I think that if you want to commercialize, you would need a novel or new compound. There's no big company that will invest in an old compound unless it is for the purpose of repurposing it. Then that is use patent, not a composition of matter patent, as Dr. Anne Villalobos is very familiar with. So those are some concerns. And I think that in the end, what's great about Doralyn's work is, okay, you reported the carbazoles, and I had told Doralyn about this in our discussions. It is actually widely occurring type of chemical scaffold in plants and in soil and in aquatic and marine organisms, and now, of course, in microorganisms. And it is a product of mixed biosynthesis. So it's got nitrogen, and then it's got the the acetate, pyruvate, fatty acid starting materials. So, and then what you reported is a halogenated carbazole. So look at the literature, and there's many attempts to chemically synthesize the more novel carbazoles. But now you have elucidated the biosynthetic pathway. So are you aiming for heterologous synthesis, like putting this gene cluster in, say, an expression system like E. coli that can grow easily in order to biosynthesize it? Okay? I think that's a very, very good point. In the case of the anthracyclines that you also reported, well, we know there are many anthracyclines that are drugs, okay? And actually, it came from the, am I right? Adriamycin came from a streptomyces from the Adriatic Sea. That's its name. That's doxorubicin. So, Doralyn, I think, if you're going to build a biosynthetic capability hand in hand with chemical synthesis capability, then like many other drugs, the more complicated structures, it's actually a combination, right? You have one part that is biosynthesized and then the other part that is chemically synthesized, and then you put them together. Anyway, so I'm sorry, it's so long, but after this, I have to go. So I just wanted to tell everyone that this has been a great webinar, and we have to be on the watch for Doralyn. Thank you.
Thank you, Dr. Concepcion, for that comment. Yes, our plate is kind of full right now. Like, are we looking for heterologous expression on this? What are we going to do with other things? So we have Balik scientists who will join our lab this year to look into the halogenase, getting the halogenase from this actinomyces, and look into what if to look into the catalysis of chlorinating other types of compounds that are metabolized by the streptomyces, and also looking into some heterologous studies. And so that is kind of exciting in our part because none of this halogenase work has been done, particularly in that on that type of halogenase that the Tubatahanensis isolate is producing. So yes, we are venturing into that. That's why we um, we have a lot of balik scientists in the pipeline who will join our group to help us elucidate more on how it is formed, optimize what compound will be the best compound, do the tweaking, should there be hit. And so parang lumalaki na po yung aming lab, which is very, very nice too dahil at the moment, it's only me and Dr. Saludes who are the lead scientists in the lab. And wala pa kaming mga iba pang mga scientists joining us. So their expertise will be complementary for our work, and that can also bring other areas of drug discovery in the pipeline.
Thank you. Do you have time for more questions? I think we can go on to the next one here from Dr. Nacario. Do you have plans to optimize your hit lead compounds through chemical synthesis prior to preclinical studies?
Okay, very good question, Dr. Nacario. Yes, na-remember ko yung lab namin sa States at sa UCSD at sa UBC. Next to the narco product lab is the synthesis lab, wherein you do all these chemical synthesis. That is in the back burner. I still believe that at the moment, hindi tayo makagawa ng like, synthesis. When I say synthesis, creating a carbon-carbon bond in order to to create another ring, or to place another functional group to make it more effective or less toxic. Dahil dito sa Pilipinas, although private school kami, we have an edge for procurement, but the lead time for the chemicals to go to the Philippines is wala 6 months pa rin or 3 months. So if you're doing your carbon-carbon bond formation, you need one chemical, or kung meron kang isa ding chemical na kailangan, kulang pa pala, so you cannot proceed. So yan yung isa naming tinatawag na we will put it in the back burner, and should there be synthesis, yes, we could collaborate with other laboratories outside siguro ng Pilipinas dahil mahirap, but it is in the plan to do some chemical synthesis for preclinical and clinical studies in the future.
Dr. Completo, would you like to chime in?
Yes. Hi, Doralyn. Congratulations again. I actually wrote my question, and it's really related to Ruel's question. So yes, so based from your answer, you are not doing synthesis because you mentioned before during your presentation that you're doing SAR. So that means that's just computational, maybe the SAR that you're trying to do. And
oh no, when I say SAR, because right now we have a compound library of different types of
okay, so based on the library.
Yeah, based on the library of what we isolate.
Oh, I see.
We do the SAR.
And then are you doing also Almitox? Because you show that your lab is capable of doing Almitox.
Correct. So right now the DOST-PCHRD has an Admitox program made up of 5 different projects. And we are one of the project looking into metabolism and toxicity using multi-imaging mass spectrometry. So the other 4 projects for animals and for distribution and absorption is done by other groups. So meron na po yung Philippines ng Admitox program.
Okay, yeah, thank you. We will contact you. I'll contact you na lang by email, and then yeah, maybe we can collaborate with our we have overlapping projects. And I saw your your project with pineapple, right?
Yes.
For lowering of cholesterol. Okay, so we
Yeah.
also we maybe we can combine our project, and we can come up with a combined product with our with our extract and your pineapple extract as an for lowering of cholesterol. So anyways, we'll contact you. Congratulations again, Doralyn, for your accomplishment. Very inspiring. Okay.
So I missed this one. Let's just ask this one. Congratulations from Dr. Peña. Congratulations, Dr. Doralyn, for your work. Any the directions towards drug formulation of your discoveries? Maybe we've already touched that a little bit, but she is happy to collaborate with you from the UPMNH Institute of Pharmaceutical Sciences too.
Yes, yes, Dr. Peña. Yes, for sure. We will contact you, particularly with formulation using functional foods that we in our project. Marami po nasa pipeline. So we will contact you.
Actually, Doc, can I ask one question? Yeah. I'm doing have you had looking at have you seen compounds that have similar structure towards docetaxel in chemotherapy? Because that's I'm doing work now on prostate cancer, and docetaxel is one of the cornerstones of treatment of prostate CA. And another drug that's very expensive is aribiterone, enzalutamide. These are very novel hormonal therapy drugs. I was just wondering about the cancer pipeline that you were going through. Which ones are the more promising leads that you have for it?
Okay. So that's a very good question. Meron kaming mga purified compounds right now that has activity on prostate cancer. However, hindi pa namin nakikita how the molecule looks like, or very similar to the molecule that you are looking into. So we know at the moment that it has an anti-proliferative activity. So we're looking into the apoptosis. What pathway of apoptosis does it target at the moment? So yan yung ang ginagawa ngayon sa aming prostate cancer. And FYI, yung prostate cancer cells na ginamit namin sa battery of anti-cancer cells, the prostate cancer cell has a very low hit when it comes to extracts that targets the prostate cancer. So parang mahirap siya patayin. I mean, I do not know. I'm not a cancer biologist. I do not know about the morphology and how this prostate cancer cells resist some toxic compounds from produced by actinomyces. But that is something that we are currently working on.
And then one last thing. You seem to be focused on staph aureus, right? Have you tried looking at, like, for example, well, at least in PGH, the number one killer really is Acinetobacter baumannii. It's been a terrible problem. Once patients get into septic shock, they get ABAO, respiratory infection. It's almost like a death sentence. And so with rising costs of care, we literally just watch them go. So have you had like work on trying to explore ABAO bacteria? I mean, other pathogenic bacteria. And ABAO is not really BSL requiring that much.
Yeah, so that's a very good question. We have 3 gram-negative bacteria that we're looking right now that are multi-drug resistant. One is Acinetobacter baumannii, Klebsiella pneumoniae, and Pseudomonas aeruginosa. And we currently have a lead. No, hindi ko ma-reveal sa'yo, sorry, on that. And we are very excited on this because, it kills that Acinetobacter on the Petri dish, on the broth, on the flow cytometer. So it is something that it is kumbaga, nandyan na. So, yung ginagawa namin ngayon is we are mass producing the actinomycin for fermentation in order to isolate that one compound. So we already have an idea what it is. So we have to make sure that we come up with a big amount so that yung structure elucidation, we will get that unambiguous. So para maalam na. But we do have Acinetobacter baumannii.
Thank you. So I think we're about to wrap up the session. We've exhausted the questions. Dr.
Okay.
Cabalan.
Okay. Well, thank you, Jericho. You know, when we started this webinar, I was just trying to explore what we have in our country. But I tell you tonight, I'm very, very proud. I am so happy. We have heard Dr. Coelho thinks about all these research centers in the country. I think San Agustin is already one of them. So we are actually over an hour from the 1 hour, but this is so interesting that I think we should go on until we can hear everything. I noticed that the budget of ASE is only $14,000. But who cares about $14,000 when we have scientists all over? They're worth $1 million. So that is why we are having these conversations. We are moving forward. So we have here a certificate of appreciation to Dr. Doralyn Dalisay, our President Mariano Santo Domingo, and Vice President Cherise Completo. Thank you very much for presenting this evening, Dr. Dalisay. Before I let everybody go, I would like to invite you in about 2 weeks. And we are going to focus our attention to artificial intelligence. Artificial intelligence. Oh, this is a very informal meeting. I see that the hand of Dr. Kezak Ivy Cub. Sir, you have the floor. Kabanting. Let's see what you have in mind. Dr. Kabanting, unmute. Oh, maybe he's gone. Well, we'll have plenty of opportunity to converse, but I would like to invite you in about a couple of weeks. We're going to pay attention to artificial intelligence. Today, I asked ChatGPT to write a poem in Tagalog. Ano? Tignan natin kung marunong mag-Tagalog itong ChatGPT. So I said, Chat, could you write a poem on a young person wanting to be old? And then when an old person wanting to be young. So it did put a poem in Tagalog. I was very surprised. But we have Dr. Prospero Naval, Professor of Computer Science from Diliman, and he will present on the use of artificial intelligence in healthcare. I saw a presentation of Dr. Naval in December where the computer could diagnose X-rays. You do not have to have a radiologist, I believe, anymore. But there are other opportunities here. Here we're looking at the molecular biology of cancer and all these can they be predicted by artificial intelligence as to what the chemical structure that it should be to probably get rid of that at PGH. So I can talk, I guess, all night, but I want to thank everyone who is present tonight, especially Dr. Doralyn, our presenter. Thank you very much again, Pangase, for staging this platform. President Santo Domingo, Vice President Completo, our inspiring leader in research, Dr. Gisela Concepcion. Thank you very much. Good evening from the United States. And thank you again, Dr. Dalisay. Thank you, Jericho, for having us.
Excuse me, can we have a picture, please?
Oh, picture. Marunong ka ba mag-picture? Ako, hindi, I could not present my introduction. Pwede ba?
Yes, Feb, are you still here? Can you please open your videos so that we can take
Everybody open their video.
Hold on. Thank you. I rarely see Doralyn. We should document this.
Yeah.
1, 2, 3. Smile. Another one po. 1, 2, 3. Smile. Done. Thank you.
Thank you very much,
Thank you, Feb, for helping us tonight.
Yes, thank you.
Bye-bye. Thank you.
Thank you all.